So glad that you came to join us for Origins, the Evidence, an exciting look at faith and science and how the two come together and the questions that you find at the intersection between faith and science. We're back for our second presentation, and our presentation this afternoon is the God Code, DNA discoveries that challenge evolution. And if you missed the first presentation, don't worry. It was all recorded and is available to view on our live stream church web YouTube channel. And if you missed your handout from presentation number 1 and you're here in person, you'll be able to get a handout from the very first presentation. And of course, each presentation, after we've finished producing this whole series, you'll be able to scan the QR code on the back of the workbooks and pull up the videos to be able to watch. So these workbooks are going to be invaluable for you, and I hope you really enjoy them. I think it's a great deal to be able to have those for free. And speaking of which, this entire series is free, but it did cost something. Believe it or not, these beautiful workbooks did cost quite a bit for printing. If at some point you are inclined to donate to help offset some of the costs for this seminar, please do. We'll have an opportunity for that tomorrow afternoon if you'd like. No pressure at all. This is completely free, and we're just glad you're here. So before we begin our presentation tonight, shall we go ahead and start with prayer? Can we do that together? Let's bow our heads. Father in heaven, thank you so much for the privilege that is ours to be able to talk about this natural world, the creation, and evidence for that. I pray that you would bring faith tonight. I pray that you would bring understanding and clearness this afternoon as we go through this presentation. I pray that you can lead and guide us. We ask it all in Jesus' name. Amen. Oh yes, one other thing I wanted to mention is that if you aren't wanting to just do the free option of the YouTube videos online, we should have DVD sets of this entire series as well at the end of the series. So this series looks at questions you find at the intersection of faith and science. Questions like, does God exist? Where did life on planet Earth come from? Can Charles Darwin and his theory of evolution explain the origins of life? Origins and tonight's presentation look at these big life questions through the lens of science. But before we look into what the book of science says, I want you each to see what the book of God says, the Bible. And it says here, and I notice that the Bible's claim is that we should be able to look into the natural world, up into our universe and down into our own life-filled planet, and find the fingerprints of God left behind in every star, every tree, on every leaf on every tree, every cell in your body. Every part of creation should contain the signature of God. Romans 1:21, read it with me. What does it say? For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead. When we look at nature, we should be able to see the fingerprints of our Creator. In Psalms 19 verse 1, it says, the heavens declare what? The glory of God, and the firmament shows his handiwork. So last night, in our first presentation, we discovered God's signature in our universe. We began with the discovery that our universe is expanding in every direction. Galaxies are drifting further and further from one another each and every day. Logically, if you go back in time, the universe would have been smaller, everything closer together. This information has led many scientists to conclude unavoidably that our universe must have had a beginning. Our universe can't be eternal. Next, we looked at conclusive evidence that the Big Bang cannot explain. Evidence such as moons and planets and whole galaxies rotating in both directions. Evidence of mature galaxies and mature clusters at the edge of space, a place where only stars should have been forming in the evolution of our universe. And finally, evidence of stars supposedly older than the universe itself. You remember the name of that star? What was it called? The Methuselah star. And if you're not familiar, Methuselah was the oldest person recorded in the Bible. That's why it's called the Methuselah star, older by nearly 1 billion years than the Big Bang model projects. We concluded our presentation together by looking at the second law of thermodynamics, which says that all energy in our universe is being irreversibly used up. So if we lived in an eternal universe, there would be no useful energy left. And because there's useful energy left in our universe, we know that we don't live in an eternal universe. We lived in a universe that had a beginning. Same thing with the second law of thermodynamics also says that we're moving gradually towards chaos. Everything is falling apart. We don't wear the same clothes we did 40 years ago. We don't drive the same cars we drove 40 years ago. Well, maybe you do, but yeah. But I'm sure there's a few tune-ups you've had to make, right? So if we lived in an eternal universe, we would be living in a universe without usable energy and a universe where everything would have fallen apart. But because we witness a relatively young universe with tons of unused energy and order everywhere we look, we can know that our universe is not eternal, but it had a beginning, a point in time when matter, energy, and space were created. And the only worldview that fits that is theism, a worldview that is in complete harmony with the Bible. A universe with a beginning. So today, we're going to move from outer space down to our own planet to look at the evidence right here pointing to an intelligent designer responsible for the origin of life. Now, up until this moment in scientific discovery, Earth is the only place in all the vast universe known to contain life. There are questions right here that need an answer, and the most central question is, where did life on planet Earth come from? And we're going to look together at the evidence from God's second book, Nature, to discover the truth that proves that the Bible was right when it said all life only came from one source, a loving God who purposefully designed everything we see on this planet. But we're going to begin with the story of Charles Darwin to see how the theory of evolution was developed, because that is the theory that competes with the Bible's proposition of creation. The theory of evolution is the competing theory that says, no, life came from a different source. And that theory started with Charles Darwin. Who is Charles Darwin? Charles Darwin, at the age of 22, had the opportunity of a lifetime. A round-the-world voyage on a ship called the HMS Beagle. This remarkable journey not only changed Darwin's view of the world, but shook science as nothing else had. Once he got on the ship, he was handed a book called Principles in Geology, written by a geologist by the name of Charles Lyell. Lyell's theory said that rocks were millions of years old. Canyons took vast amounts of time to form. And all of this Lyell called deep time. Darwin had 2 months on that voyage, rocking back and forth to read that book. Now, I don't know about you, but I can't even go in a car and read, let alone go on a boat and read. But 2 months, that was the book that he read. And by the time the ship had landed in South America, he was fully convinced that Lyell was right. Everything that we see around us had to have taken an extremely long time to form. There was another influential book that Darwin read on his voyage, a book written by his own grandfather, Erasmus Darwin. This book claimed that all animals were related to one another. They were all connected somehow. And this thought had a tremendous impact on Darwin. So as the ship traveled around South America, charting the coastline, Darwin sought to explain everything that he saw through the deep time theory he had learned from Lyell. This proved pivotal when the boat finally landed among the Galapagos Islands. There, Darwin noticed slight changes between mockingbirds on the islands and those on the mainland. This was revolutionary for Darwin, for up until this moment, he had held the belief that species cannot change or adapt. They're fixed. There's no possibility of them changing, which he had learned from reading Lyell's books, and also this was the cultural belief at the time, that all species were fixed. They couldn't change or adapt at all. Darwin also noticed that the finches on the different islands had different sized beaks. So with the lens of deep time provided by Charles Lyell and the lens of his grandfather's book Zoonomia, which implanted the idea that species were all related, Charles Darwin began to put all of these ideas together, and he came to the belief that everything must have a common ancestor. Once Darwin was back from his voyage, he began to develop his theory. And here's how his theory goes. Over vast amounts of time. Where did he get that from? Well, that was from Lyell. Guess I needed to put that arrow over there. Small and slight changes within species. Where did he get that from? The Galapagos Islands. Explains how all species are connected. Where did he get that from? His grandfather, Erasmus Darwin. And now comes the new thought, through a common ancestor. So he put all of these ideas together, and this is how Darwin began to formulate the theory of evolution. The link was now made, and Darwin published his book, Origin of Species, November 1859. In Darwin's book, the illustration of an evolutionary tree shows how all living things, plants, fish, animals, and humans, trace their common ancestry back to a single life form. Everything came from one single life form. Darwin's theory was an attempt at explaining the entire world and the diversity we see in it from one original life source. He wrote, all organic beings which have ever lived on this earth have descended from one primordial form. That's in his book, Origin of Species. With this idea, Darwin had a theory that explained the earth, but there was one major problem. How did that first life form, that primordial form, come into existence? That first original cell. If God didn't create the first life, then where did it come from? Darwin sought to explain everything through natural means. So in Darwin's mind, the first life had to have come from what? Non-living things that existed before it. It had to come from rocks or dirt or non-living matter. Whatever was on this planet before the first form of life was the stuff that life came from. So the haunting question for the theory of evolution was really, how do you get life from non-living matter? The entire theory of evolution was based upon slight changes over long periods of time. Therefore, if the first life form could not be explained through slight changes over a vast amount of time, the theory of evolution would fall apart. How did that first life here come from non-living matter here? This is how Darwin put it in his own words. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, what does he say? My theory would what? Absolutely break down. And this is what a real scientist does. They give you a way not only to prove their theory, but a way to disprove their theory. And so according to Darwin, the way that we disprove his theory is to show that any complex organ, that's any animal, any plant, any cellular, mono or multicellular organism, could not have formed through numerous successive slight modifications. So let's take a look at how scientists have sought to explain the origin of life from non-living matter. Now, the origin of life from non-living matter is called chemical evolution, or the more technical term is abiogenesis. It's an attempt to essentially describe how life could come from rocks or anything that pre-existed it. And this idea actually predates Darwin. You can go all the way back to Aristotle. Aristotle and ancient Greek philosophers. According to Aristotle, everyone could observe that the tiny insects called aphids, they came from raindrops. Flies spontaneously popped out of rotting food. Mice, you know where mice came from? Dirty hay bales. And everyone knows that rotten logs produce crocodiles. This theory of Aristotle was later disproved by Antony Leeuwenhoek in 1680. With technological advances in science, by the 1800s, scientists thought that life must have come from an original single-celled organism, which was easy to imagine because a single cell at that time was just a globule of plasma, or a plasm, like a dollop of jello on your plate. All you needed to do was explain how a wall formed around that jello, and you've got your first cell. Today, we understand that explaining the existence of a cell is not that easy. You need much more than just a cell wall to have a living cell. Cells are made of literally hundreds of different proteins that all carry out essential functions within the cell. Without proteins, a simple cell could not exist. Literally, life depends upon protein. No protein, no life. And we're not talking about eating protein, we're talking about what you're made of. Every part of you is made of protein. Darwin must have understood that proteins were the key to understanding the first life, because in a letter dated February 1, 1871, Darwin suggested that it was in a warm little pond with all sorts of ammonia and phosphoric salts, light and heat, and electricity that a protein compound was chemically formed, ready to undergo still more complex changes. And Darwin was right on this point. Scientists today agree that protein is the one substance necessary for life to evolve. So the challenge of evolution is describing the evolution of protein. If you can describe how protein formed by chance, then evolution has a chance. If protein cannot form by chance, then the evolution of the first cell and the evolution of all life is absolutely impossible. The entire theory of evolution hinges completely on the evolution of protein. So what exactly is protein? Protein, or proteins, are little three-dimensional machines or structures that carry out hundreds of necessary jobs in the cell. And I want you to think of proteins like a toolkit. What sort of things do you have in your home to help repair your home? Something breaks, you have a hammer, right? So you pull out a hammer. What's a hammer used for? To pound in a nail. Is a hammer very good at screwing in screws? No. I mean, if you hit it hard enough, maybe you can pound in the screw. But how about, is a hammer good for tightening bolts? Not at all. Loosening pipes? Well, maybe. Sawing a board in half, is a hammer good for that? No, you'd be sitting there beating it till you're blue in the face to get that board to break in half. That's what proteins are. Proteins are three-dimensional structures that are in a specific shape for a specific job. So each protein has its own specific function, and the shape of the protein is its function. And you need hundreds of these proteins in a cell in order to carry out all the functions in the cell. Some proteins are like the bones in our body. They give the cell a specific shape. Others are little machines that speed up reactions in the cells. Those are called enzymes. They go at lightning speeds. Still others transmit signals, aiding communication in the cell. Some can transport waste and nutrients in and out of the cell. Still others act like an army, binding to foreign invaders in the cell to protect it. Proteins are not only incredible, but essential to every cell. So without proteins, you cannot have a functioning cell. Just like without tools, you cannot build a habitable house, right? You need the tools in order to function. So what do you need for a protein to form? Well, proteins are made up of tiny molecules called, does anybody know? Amino acids. Do you know how many different types of amino acids there are? 20. 20 different types of amino acids. And each one of those amino acids, if you can just imagine them as little blocks, colored blocks, those amino acids line up in a very specific sequence to form a protein chain. So if I labeled the amino acids by color, a specific sequence of amino acids to form a protein might look something like this. You have a red one, and a blue one, and an orange one, and a green one, followed by another blue one, and a red one, by a purple, and yellow, and then green. And only if it's in that sequence are you able to have a protein. If the sequence is mismatched, the protein isn't going to function correctly. So you have this sequence. If we put it into numbers, it might make a little bit more sense. If you have 20 available amino acids to put in a chain, you can view them as a sequence of numbers. So you have amino acid number 1, followed by amino acid number 5, followed by amino acid number 20, and so on. If you have one single amino acid missing or put in the wrong place, the whole protein structure falls apart. The average protein chain is 300 amino acids long. So here's the question. What is the chance of 300 amino acids lining up perfectly in the correct sequence to form a single simple protein, just one protein? Here's what you need if chemical evolution is going to work. Number 1, you need amino acids. So you must prove that amino acids can form from chemicals. Number 2, you need amino acids to connect together somehow, like magnets connect. Number 3, you need those amino acids to line up in a specific sequence. Number 4, you need those amino acids not only to line up in a specific sequence, but then they have to be folded into the correct shape or they're not going to do anything. And then number 5, you need a whole bunch of different proteins all correctly folded into all the necessary structures needed to run a single living cell, and only then do you arrive at the starting place of evolution. So evolutionary scientists have been seeking to work this problem out. In 1953, Stanley Miller tried to simulate Darwin's warm little pond where the first protein was supposed to have been formed. He hoped to form amino acids from chemicals by blasting and zapping the chemicals over and over with electricity, simulating lightning strikes. And it was amazing. After 2 weeks, the methane, ammonia, hydrogen, and water in his experiment produced 13 out of the necessary 20 amino acids needed for a protein. 13 out of 20, not too bad, just over half. It was hailed a scientific breakthrough. But did this close the case on how proteins are formed? Not yet. Miller had accomplished the first and the easiest of all steps. The more challenging problem was to get those amino acids to connect to one another. None of them did in Miller's experiment. No amino acid chains formed, no proteins were created. The closest that science has ever come has been to artificially produce 13 amino acids by zapping gases over and over and over in a container. This only proved that amino acids could form if an intelligent being purposefully tried to create them in a perfect environment. But what about in the wild, where conditions aren't perfect and you don't have an intelligent being in a lab trying to create amino acids? And then you still aren't able to explain, if they did form in the wild by chance, how did they connect together to form a specifically sequenced protein chain ready to be further folded into the current shape needed to be able to function in the cell? Dr. Dean Kenyon tried his hand in an explanation when he said, life might have been biochemically predestined by the properties of attraction that exist between its chemical parts. particularly between amino acids in proteins. Essentially, he's saying, you know, maybe what happens is amino acids just kind of connect together without anybody forming them, without any sort of direction. They just kind of line up together. You know, like if you were to roll a bunch of magnets on a table, they would all kind of attract together, and all of a sudden, whoop, you've got a whole chain. So this is what he published in his book, Biochemical Predestination. And then one of his students who read his book came up to him and said, Dr. Kenyon, there's nothing in all of science that shows that your proposition is true. The only way that amino acids connect together to form a protein is through DNA. The DNA in every living cell, whether it's the cells of a grasshopper or the cells of a Einstein, all of them have DNA that direct the forming of proteins. We're going to go over protein formation here in a little bit. It will be really exciting. You'll fall out of your seat, I'm sure. So Dr. Dean Kenyon, he took a look at it and he said, you know what? I was wrong. The only way that proteins can form is through DNA. Random chance can't do it. And so Dr. Kenyon reversed his opinion and has since rejected his own book, Biochemical Evolution. He now believes that the only way that proteins can form is through DNA. It's like trying to come up with the right sequence by guessing the numbers in a combination lock. How many of you have tried to guess the combination out there? Oh, you greedy people. No, there has been one person who has actually guessed the numbers, and they guessed it last night. My son, after all of you left, was there trying to guess all the numbers, and he actually guessed the numbers. He came to me with the open box. He said, Dad, look, I guessed it. I said, shut the box and don't tell anybody the code. So it is possible, but it's going to be difficult. So let's say that you had a combination lock and there's only 3 or 4 places, each with 10 numbers to guess from. In proteins, you don't have 3 or 4 places. You have 300 or 400 different places in each place. And instead of 10 numbers to choose from, you have 20 numbers in each one of those places to choose from. So what are the odds of a protein forming in the wild through a random, undirected process? Let's just call it by chance. What are the odds of a small protein, 150 amino acids long? Well, Doug Axe said a short protein chain is made up of about 100 to 120 amino acids. And just to put that into perspective, the longest human protein chain is titin with 34,350 amino acids, all in a correct sequence. That's a long chain, long protein chain. And here it is on the screen. That's titin right there. 34,000. So we're a long way away from that by just going with 150 amino acids. If there are 20 possible amino acids and you could form a protein chain that was just 1 amino acid long, what are your chances of forming that chain? Just 1 amino acid, picking the right one, and you've got 20 to choose from. What are your chances? 1 in 20. After 20 guesses, you're very likely to get it right. How long would that take you? Well, Stanley Miller's experiment showed that 13 amino acids could be produced in 2 weeks. So it shouldn't take more than a month, perhaps, to come up with a single right amino acid. However, we don't have to come up with just 1. We have to come up with 150 to sequence a small protein, or 34,350 if you want to sequence the largest protein chain. So what are the odds of a protein that's 150 amino acids long? Any idea? 1 times 10 to the 195th power. And remember, we're not talking about titin, the longest protein chain. We're talking about a relatively small protein chain at 150 amino acids long. To put that into perspective, In the whole known universe, there are only an estimate of 10 to the 80th power of elementary particles, protons, neutrons, and electrons in the whole universe. There is a greater chance for you to find a single marked proton or one specific electron searching the entire universe than for one simple protein chain to be lined up correctly by chance. Now let's say that for every second on a watch, a new amino acid combination was tried. Every single second in the primordial soup, a new combination of amino acids was tried. According to probability, you would need 1 times 10 to the 195th seconds. If the universe is 14 billion years old, you would have to have a total of approximately 10 to the 17th seconds in the total universe. You don't have enough time, even from when they say the Big Bang began, you don't have enough time if you tried a new combination every second to come up with one single protein. And to add to that, there are 30,000 different proteins, and most of those proteins average an amino acid chain that is double what we just looked at. The average protein is 300 amino acids, all correctly sequenced long. So there isn't enough time even if you used every second. Coming up with amino acids was the first problem. And since science has proven this can happen in a perfect environment. The second problem in forming a protein is sequencing them all right. And science has proven this cannot happen even in a perfect environment. The third problem is getting the amino acids to bond correctly. To form a functional protein, amino acids have to form a specific bond called a peptide bond. A peptide bond only occurs in 50% of all the bonds that happen between amino acids. Without this peptide bond, a protein cannot form. In a protein that requires 150 amino acids, you have 149 bond sites. So you got, 2 chances with each bond site to get it right. That is 1 in 10 to the 49th power chance of just getting all the bonds correct for a single protein. And not only that, but amino acids, each amino acid comes in 2 forms, a right-bending and a left-bending amino acids. Proteins can only use left-bending amino acids. So all of the amino acids in the chain have to be left-bending in order for them to work. Again, you've got 1 times 10 to the 49th power of that happening. And I know that these numbers are so big, you're going to be dreaming about this tonight. Amino acids must form from chemicals. Amino acids in the correct sequence is 10 to the 195th power. And then you have amino acids must form correct bonds. That's 10 to the 49th power. And then you have amino acids can only be left-handed. The chances of that is 10 to the 49th power. In the unlikely possibility that all these things could happen and a single protein does form, you still haven't arrived at the place Darwin began, because it takes between 250 and 400 different proteins to form a simple single cell. Are you beginning to see how impossible this is for chance to explain? It's one of the greatest challenges to Darwinian evolution. The only way that this can happen is if you have enough time for random sequences to occur, and the entire universe doesn't have enough time. This is a problem for evolution, but my friends, it isn't a problem for an intelligent designer. It isn't a problem if God created each protein, each different cell, each life form, and then coded it all to happen. So what do we observe in our natural world? How do proteins form today? What is the only way that scientists who use the scientific method have observed proteins being formed? Are they formed by random chance? Far from it. Far from it. Scientists today understand that proteins don't happen by chance. Everyone knows about DNA, right? DNA contains a very complex code, more complex than any computer ever built. This code has all the information needed to manufacture proteins. So how does it happen? Well, it all happens right here in the nucleus of the cell. And if we zoom into the nucleus of the cell, what we find is that there's a protein. There's the cell nucleus, and inside the cell nucleus there's DNA, and it's all coiled up like a gigantic scroll, just waiting for someone to read it. And when a protein is going to be made, a machine called RNA polymerase, which is itself a protein, unwinds the exact portion of DNA where the instructions on how to build the specific protein are contained. So it goes through the library, and it picks out the books that it needs to read. Okay, let me find the recipe for the protein I need. Oh yes, right here. And it can't take the book with it. It has to photocopy the book because DNA remains in the nucleus of the cell. So it takes a photocopy of that book, and the photocopy is called RNA. And it takes the RNA, and then another protein molecule transports that RNA out of the cell. So the RNA travels out of the cell nucleus into the outer layer of the cell, where it finds another protein machine called a ribosome. The ribosome is like a factory that will assemble the protein chain. It's kind of like the kitchen in your house. Somebody goes downstairs, they get the recipe book, but they can't bring the recipe book upstairs, so they photocopy the recipe, and then they hand it to their brother or sister, and the brother or sister is the one who carries the recipe upstairs. They finally get upstairs and says, okay, Mom, here's the recipe. The recipe is all the ingredients that need to come together to make that protein chain. The cook is this ribosome factory. So it takes the RNA right here, the recipe, it goes into the ribosome factory, and the ribosome factory says, okay, I need all of these ingredients. And there's another little protein called tRNA enzyme, and tRNA enzyme goes and scavenges all of the amino acids needed in that recipe, and it brings it back to the kitchen. And in the kitchen, the ribosome factory now begins to assemble each amino acid according to the code that was written or copied in the RNA. And the code came directly from the center of the cell, the nucleus. So it says, okay, I need amino acid number 5, followed by amino acid number 7, followed by amino acid number 20, followed by a 2 and a 2 and a 3 and a 20, and it continues to put them all together. And then once all of the amino acids have been put together, you have what's called a protein chain. They're all fit together. Now, it's not complete yet because the protein chain still needs to be carried out from the ribosome factory to a new factory called a chaperonin. A chaperonin is another protein structure that's like a barrel. This is like your mother taking the pie that she's just made and saying, I need to cook it in the oven, right? So the chaperonin takes this protein structure and begins to fold it. And it folds it and folds it and folds it and folds it until the protein is folded into the exact shape that it's needed. And only then is the protein then carried into the rest of the cell to be able to function. That whole process is mentioned in the workbook. So if you need to, you can just read that over and over tonight. Round and round. It works better than a sleeping aid. So there's the protein chain, there's the folded protein, and that's how it works. Now, some of the proteins that are in a cell are called enzymes. Enzymes are just incredible. What would take a cell literally millions of years to accomplish without an enzyme, with an enzyme can happen in seconds. Enzymes help speed up cellular activities to unimaginable speeds, 1 × 10 to the 18th power. That's how amazing they are. Enzymes are specific tools made to speed up the job. The same jobs without them would take millions of years. Enzymes are necessary to read DNA. What is necessary to read DNA? Enzymes. But what is necessary to make enzymes? DNA. In order to read the DNA to make enzymes, you need an enzyme. In order to have an enzyme, you need the code in DNA. This is kind of like which came first, the chicken or the egg, right? Do you see how both of them need to exist together in order for either one of them to be usable or to exist? Without DNA, you cannot have life. We call this irreducible complexity, and I'll be covering that in the next presentation a little bit more. You have to have the instructions in DNA for life to begin. Bill Gates, founder of Microsoft, said human DNA is like a computer program, but far, far more advanced than any we've ever created. Berland Olaf Coopers, a German biochemist, says the problem of the origin of life is clearly basically equivalent to the problem of the origin of biological information. Without information, you can't have life. So let's look at the information in DNA. There are 3 essential elements for DNA to work in the cell. There needs to be a language or a code. Information cannot be shared unless you have some language to share it. I want you to think of how we share information with one another today. We always use a language, right? English, Spanish, Chinese, or we can use a language like computer programming. Language is simply a logical, sequenced code that conveys information. In DNA, this sequence is a 4-letter code, A, G, C, T. If you think of our modern computers, they run off of only 2 letters, called a binary code. How much information can you store on a computer with just 2 letters? A, B, A, B, A, B. How much information? All the information in the world stored right here, just using 2 letters. How many letters does a DNA spiral have, helix have? 4 letters. Not 2, 4. In the same transcription of DNA, you can have multiple different instructions all layered on top of each other. It's incredible how much information can be stored in DNA. Far more powerful than any computer we know today. So there needs to be a language. There needs to be a communication channel as well. DNA communicates through its channel RNA. And then there needs to be meaningful information. Whatever information DNA passes on needs to have a purpose. It needs to be readable. It needs to be meaningful. So how is the language in DNA developed? Why is there highly specific information at all in DNA? And where did the information in DNA come from? Well, first, the information in DNA has nothing to do with the actual physical DNA structure. And I'm going to illustrate this to you. I want you to look at this piece of paper. And then I want you to look at this piece of paper. What is the physical difference between these 2 pieces of paper? Physically, both of them are made of paper and ink. Right? They both have no physical difference other than this ink was laid out in a very orderly, specific way. The information on the paper is completely separate from the structure of the paper, from the material of the paper. Does that make sense? Are you following? So ink and paper does not make information. You need something more. You need intelligence to arrange the ink on the paper in order for it to actually convey information. So the chemical components of DNA do not explain the information in DNA. You'll never be able to explain how the paper and ink in a book chemically evolved and then use that chemical evolution to explain Shakespeare and how it was written. Information cannot be explained by chemical evolution. Information has only one source: an intelligent being. Consider the SETI program. NASA has a program that scans outer space looking for signals, codes, anything that may be from an alien. What would happen if scientists suddenly picked up a signal from space with the simple Morse code SOS? What would scientists automatically say? Without question, every major newspaper around the globe, every major news site online, splashed across all social media accounts would be the story, aliens have contacted humans. We've finally got a message from outer space. Now, what about here on Earth? What happens when humans discover information like that? Here's an example, petroglyphs. In Arizona and other places, you'll sometimes stumble across pictures on rocks that look like sheep or stars or humans or other objects. When we see those pictures on rocks, hold your breath. Okay, hopefully it stays. When we see those pictures on rocks, do we look at them and say, you know what? I bet 1 million storms with 1 billion raindrops produced those engravings on that rock. Over long periods of time, they just happened to arrive there because of the raindrops splattering in this random pattern. You know, when we see design, our automatic thinking is there must be a human, an intelligence, something that made that. Even if we've never met the human, seen the human, or know anything about the human, we automatically assume when we see petroglyphs that there is an intelligent designer. When a hiker or even a scientist with his PhD automatically or sees these petroglyphs, they automatically assume that a tribe of Indians must have painted them on the rocks. And we automatically assume that they came from an intelligent source. Now, why is this not random chance? Why is it not possible that these paintings just evolved? Why do we assume it came from humans that we've never seen or met? Because we all intuitively know that without intelligence, you just have randomness. And randomness does not produce information. Books, videos, CDs, even paintings in museums all have an author, a creator, and an intelligent designer. DNA contains a more advanced information than anything we've ever built or designed or made. And that information in DNA that is absolutely necessary for a working cell to exist and for any organism to have life came from an intelligent source. The DNA book was written by an author. The masterpiece of DNA was sculpted through the working of some creator. When you look at a DNA strand, you aren't just looking at ink and paper, you're looking at something that was designed. Henry Quasler, a pioneer in molecular biology, said this: the creation of new information is habitually associated with what? Conscious activity. Richard Dawkins said that the machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. Francis Crick, Nobel laureate on DNA research, said, biologists, now pay attention, biologists must constantly keep in mind that what they see was not what? Now, why do you have to constantly keep that in mind? Because it's obvious, right? The heavens declare the glory of God. The firmament show His handiwork. You have to continually lie to yourself. What I'm seeing was not designed. It wasn't designed. It wasn't designed. I'm going to brainwash myself into believing it wasn't designed. Because it looks designed. Now, what about junk DNA? You know, only 2% of the genome was used for making proteins, so scientists conclude that 98% of our DNA is junk DNA. Through the evolutionary lens, these useless genes are simply the leftover products of millions of years of evolving. This seemed to provide a huge support of evolution and a crushing blow to intelligent design until September 6, 2012. It turns out they were wrong. In an impressive series of more than 30 papers published in several journals, including Nature, Genome Research, Genome Biology, Science and Cell, scientists now report that these vast stretches of seeming junk DNA are actually the seat of crucial gene-controlling activity. Junk DNA? No way. The next crushing blow came with the discovery of genetic entropy. What's genetic entropy? Well, genetic entropy is the discovery that the information in DNA is degrading over time, like an old book that is slowly crumbling apart with age. Rather than supporting evolution's idea that new genetic information is being created all the time, genetic entropy shows quite the opposite. Instead, it says that we are losing genetic information over time. Every time a cell divides, 3 mutations occur. These are the mutations that cause us to get old and die. A portion of the mutations in each person is passed on to their offspring. Every generation begins with more mutations in their DNA than their parents. The human race is slowly degenerating. And some may say genetic mutation is good. And that's how evolution works, through gene mutation. Evolution teaches that the only way that a cell can evolve is through genetic mutations. That's what anthropologist Brianna Pobner said. She said mutations are basically the raw material on which evolution acts. In other words, if one creature is going to evolve into another, it has to happen through mutations of the genetic code. All the evidence in science shows that mutations can only remove or alter information that's already present. And this is a really important point for you to understand. There is no scientific research anywhere in the world that shows that new genetic information is added to any living life form. Mutations never add new genetic information. Mutations only alter the information that is there or delete the information that is there. And that's true when you look at sickle cell anemia. Now, the normal blood cell is shaped like a donut, right? Or round. Sickle cell is a mutation of the genes where the blood cell is shaped like a moon. And it actually gives a genetic advantage to some people. Do you know what the genetic advantage is? Malaria. That's right. You can't get malaria. You're immune to malaria if you have sickle cell anemia. However, sickle cell anemia also comes with a whole host of life-threatening issues. Blood vessel blockage leading to anemia, low blood oxygen, increased risk of bacterial infection, increased risk of stroke. Mutations, you see, work in the wrong direction for evolution by destroying information, not creating new information. So even mutations are a problem for evolution. Remember what Darwin said? He said, can you read it with me? If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. So here's the question. Without the first cell or the first life, can Darwin's theory work? Is it possible to get that first life through numerous successive slight modifications? You could answer. There's no way. And in case you didn't catch it, here's the review. Proteins. You need proteins for life? Stanley Miller was able to produce 13 amino acids from chemicals, but there isn't enough time in the universe for those amino acids to get the right sequence for a small protein chain. This problem is compounded because you also need the right bonds and also the right bends, and then you need to fold the protein. Lastly, you need about 250 of these proteins for life. None of it could happen by chance. There just isn't enough time. We know that proteins are formed through DNA. DNA is needed to form proteins and enzymes, both of which are needed to read the information in DNA. It is the information in DNA that is crucial to making proteins. This information cannot be explained by chemical evolution. We readily observe that information comes only from intelligent sources, and junk DNA is a myth. Our DNA is also degrading with every generation. We're losing genetic information rather than gaining new genetic information. At the very end, by looking at all of this information, we can say, Charles Darwin, I think we've disproved the theory of evolution just with looking at how proteins are formed. Where did it all come from? Intelligent design is the only theory that can currently answer Darwin's dilemma. How many today would say evolution has an impossible mountain to climb to be accepted? And if you don't accept evolution to explain life on Earth, what are you going to believe that fits the evidence? The only alternative to evolution is intelligent design. Okay. Go get water. Take a drink. We're going to take a short 5-minute break, and then we'll come back for our presentation, Nature's Marvels, where we'll uncover a whole host of creatures that defy evolution.